Hello and welcome to Inside Exercise. I'm Emeritus Professor Glenn McConell. I've just finished a really enjoyable chat with Professor Mark Hargreaves from the University of Melbourne. He was actually my PhD supervisor back in the early 90s. He's a remarkable person, very pleasant, a great mentor to me during my PhD, but also afterwards, and very supportive with all his PhD students and others. He has a remarkable track record in exercise metabolism and exercise physiology. Over 200 peer-reviewed journal articles, has been invited to many conferences as a keynote speaker, many invited reviews, has had a massive influence in the field. So we talk today about all things carbohydrate metabolism and exercise. So the effect of glycogen availability on exercise metabolism and performance, the effect of altering carbohydrate availability in terms of glucose ingestion, carbohydrate ingestion during exercise, the effects on glucose uptake, liver glucose output, and exercise performance. Also carbohydrate availability and fatigue. We also talked about GLUT4, so the glucose transporter, GLUT4 in muscle and fat. Other discussions were around fluid ingestion, so not just carbohydrate ingestion, but the effect of different volumes of fluid ingestion, the effect of addition of electrolytes or not. So I really think you'll enjoy this one, so stick around. Hi, Mark, how are you? Welcome to Inside Exercise.
Thanks, Glenn. Good to see you.
Great to see you. Thanks for coming on.
That's all right.
So some people will know that you were my PhD student, supervisor, and I was only your second student. So that was a really great experience. And I often think, when students would come to see me and whatever, how you always had an open door policy.
Yeah. Well, no, it was a good time. You and Mike McCoy and Mark Hargreaves was at another institution, and we were involved around all around the same time doing stuff on carbohydrates, and it was and we'll get on to our double tracer study, but that still is one a study of which I'm most proud, I think.
I agree. That was awesome study. And so always an open door policy, always helpful. I realized actually after a while that I can't just come in every time I have a question. So I'd save them up and come in, but you're always great. And the other thing I learned and I stuck to was that whenever you had a paper accepted or, an award or something, you'd straight away put it in your CV, rather than, having to try and remember them later. And I always did that as well. So it was a good tip. And you'd come back from Ball State, and I'd been at Ball State, and then David Costel came over from Ball State, and then Andy Coggan came for a visit from Ball State. And indeed, we've had a few Ball State people on. So
yes, there's a network of Ball State alum, which speaks to the influence that Dave and that lab has had on the field. Yes,
and I've tried to have Dave on here, but he's not. Maybe he's too modest or something. He's not. He hasn't fronted yet.
Yes. Okay.
All right. So we're going to be talking about carbohydrate metabolism and exercise, of course, which you're obviously a world leader on and a huge name in exercise physiology. So how about we just go through that, and then I'm going to slot in some questions and things I've had on Twitter. There's been a big interest on Twitter when I said you were coming on, and other bits and pieces, and some of the things we tried to think about years ago, what's going on and whatever.
Yeah.
So I don't know how you want to structure this. So going through, we're talking about glycogen and then glucose and carbohydrate ingestion, fatigue, whatever. Do you want to start with, I don't know, glycogen or?
Well, it's scary to think that it's now 40 years since I first went to Ball State, and at one level you go, gee, where did that time go? And at another level, it seems like it's ages ago. So, but when I got to Ball State, Dave had a pretty active lab. Andy Coggan was there, Abe Katz was there. And there were projects going in the lab, some of which were industry sponsored, some of which were agency sponsored. So it was a real, a wonderful baptism by total immersion, if you like. I'd done physiology at Melbourne, a little bit of exercise work with Mary Chennels on oxygen uptake in elite rowers, but nothing really too elaborate. And then getting to Ball State, we had studies on carbohydrate feeding, on alkalosis, on heat, exercise in the heat, pre-exercise carbohydrate feedings, and just a wonderful environment. And that's not surprisingly, that set the tone for me keeping an interest in exercise. And then over the years, one thing leads to another, and you end up going in interesting directions, but still broadly around the area of carbohydrate metabolism during exercise.
Yeah.
And
so
then,
I mean, so when I came down, sorry.
So I was just going to say, then obviously, Dave had links with Bengt Saltine. So I spent some time in Copenhagen, met Eric Richter, and then all sorts of other opportunities emerged from those experiences. And that was a wonderful place to do some work as well, as you know.
Yes. You've had an amazing, background, and then obviously I came along, and you were pretty fresh. You were pretty fresh from, How long since you'd finished your PhD when I came?
So I finished at the end of '88 my PhD, and then worked at Victoria University for 3 years, and then came back to Melbourne University in 1992 when the physiotherapy course started, and they were interested in having some exercise in that course. And the opportunity to come back to Melbourne, which is where I'd sort of done my undergraduate and my PhD, was very attractive. So I went back there, and sort of, I think we first had conversations when I was at VU, and then I'm, the projects I moved across. Your PhD was submitted '94.
Yeah.
So I'd been a couple of years back at Melbourne. We did most of our experiments at Melbourne. Of course, our tracer studies were with Joe Pareto, who was across the road at the hospital and had the system set up to analyze stable and radioactive glucose isotopes. So that was a good collaboration with Joe as well. So
yeah, and he'd be running across to do the muscle biopsies.
That's right. Yeah.
I said in a pod the other day how remember that time we had a guy, we were doing placebo and carbohydrate ingestion. And he went 3 hours on placebo, and then we got to about 4 hours.
5 hours or so on carbohydrate. And I think Joe ended up being parked in the office waiting for, he said, "Oh, we think he's almost there," and then he kept going. So
we actually rang up because he was across the road at the Royal Melbourne Hospital. He ran over, and then he headed back, and then he came over. So he went 5.5 hours. Now, talking of Melbourne Uni, I was just thinking about with the Australian Open on, you're a bit of a star tennis player.
I wouldn't say I was a star tennis player. That's ancient history. There's my tennis career, Glenn. So, but I played a bit of tennis growing up. Played to a reasonable level as a junior, and then decided to just keep it more recreationally and focus a bit more on the academic stuff. So
see, this is the famous Mark Hargreaves modesty. So you won people in the US and England wouldn't know this, but you won the University Blue, which is the best tennis player at Melbourne University. Is that right?
Well, I won the university championships, and that qualified me for a blue at Melbourne Uni. But most of the really good tennis players didn't go to university; they went on the circuit. So there were some good players who we played about in the university. But it was nothing like, say, the US collegiate system, or some of my junior peers with whom I played as juniors, they went and pursued their tennis careers rather than going to university. So
that's true.
Some of them went to US colleges before they went onto the circuit. So, all right, that was all good fun. So.
All right, good stuff. So yeah, if we start thinking about carbohydrate metabolism, do you mind just giving a bit of an overview of, what sort of happens during exercise, the effective intensity and duration on carbohydrate metabolism? And then we sort of get into a bit more nitty-gritty things.
Sure. So, I mean, at most of the intensities where, people who are reasonably serious about their exercise, train and compete, carbohydrate is the major fuel. And certainly, in humans, it's muscle glycogen more so than blood glucose, although the blood glucose pool is still an important source of carbohydrate for contracting muscle. And the blood glucose also is very important for the brain. And so that's an important factor during perception of fatigue and the brain metabolism, which is important. So muscle is able to use a range of fuels. Anaerobic, so-called aerobically can use triglycerides from muscle triglycerides, from adipose triglycerides, can use glucose units from glycogen in muscle, from liver glycogen, from ingested carbohydrates. So, whatever the evolutionary pressures were, human skeletal muscle is able to use a variety of fuels. And but as you increase the exercise intensity, you increase the reliance on the carbohydrate fuels, and accordingly the reduction in fat occurs. And I think perhaps one of the best one of nature's experiments is patients with McArdle's disease who lack the ability to break down glycogen. If you look at their maximal exercise, aerobic exercise tolerance, it's about 60–65% of age-matched healthy controls, healthy subjects. So there's a limit to how high you can go with exercise intensity with oxidizing fat. Now you may still have a very large stimulus for breaking down triglycerides, and indeed during very intense exercise you see increases in glycerol in the bloodstream, which reflects perhaps a breakdown of muscle triglyceride. But the utilization of that is determined not just by the availability of the fatty acids, but also the ability to get into the mitochondria. And there's good evidence that the rate of glycolytic flux influences that, as well as the availability of the fatty acids. And if you go back to the old Krogh and Lindhard studies, there's a greater oxygen cost with oxidizing fat. And teleologically, as you approach the limits of your aerobic system, it makes sense to get more value from oxidizing carbohydrate than fat, which is that's teleologically attractive. And I guess we've spent a lot of years trying to understand the mechanisms of that. So
yeah, so this is the classic exercise physiology, exercise metabolism that's in all the textbooks and things. But nowadays, I know we don't want to get into it too much, but if you ever look at Twitter, I know you're not on Twitter. Maybe this is why. Nowadays, it's just all sorts of discussion about, you should be on high fat diets and keto diets, teach your body to burn fat, so therefore you can spare your glycogen and whatever. But as you say, just the basic energetics are that if you're going at a low intensity, you can burn a lot of fat. But as it gets harder and harder, the classic studies show you switch. We don't like to talk switch, but you use more and more carbohydrate. Yeah. What do you think about this? I mean, you were involved in the studies with Louise Burke. Sure, you can teach your body to burn fat, but as soon as you try and do any sort of decent intensity, it's not going to help you.
Yeah, look, I should say I'm not a nutritionist. I'm a physiologist, and I sort of end up in this sort of sports nutrition space because of some of the work that we've done over the years. But I have no formal training in nutrition, and other than stuff I've acquired by osmosis by working with people like Louise, who is formally trained as a dietitian. And look, we saw very big increases and big changes in metabolism that were not paralleled by changes in performance always. When we would do the standard preloading at 70–75% VO2 max, then performance tests. She followed up with John Hawley and others with longer term exercise, and probably from a practical perspective, and Louise was always very interested in yes, you can do these things in the lab, but how does it make a difference for the athlete on the field? And her studies with race walkers perhaps have taken that to the real applied area. And again, looking at there's the fuel economy and there's the oxygen economy, and her studies would suggest that notwithstanding the metabolic adaptations that occurred, there were performance decrements in those race walkers, and they were related a bit to oxygen cost of exercise. And so I think there's no question that you can adapt to certain exercise intensities and burn more fat. And the very early study from Steve Finney with the metabolic changes, the nutritional ketosis that occurred, there were adaptations that allowed the subjects to exercise at 65% of VO2 max using more fat than glycogen. But we've shown, and Tim Noakes in South Africa has shown that at the higher intensities, there is a challenge to maintaining carbohydrate utilization, carbohydrate oxidation when you've been on a high fat diet. We've shown changes on PDH activity. He's suggested perhaps changes in circulating hormones, in maybe the neural factors. There's all sorts of interactions with the dietary fatty acids and some of the membrane composition that might be relevant to say bioenergetics in the mitochondria and those sorts of things. So I think it's maybe it's just hand waving to say, well, everything in moderation, but I think a lot of the extreme examples, when they're applied to context and settings where there isn't, to begin with, not a huge reliance on carbohydrate, and then there's often an excessive focus on carbohydrate. I think the analogy we were talking about briefly is with water, and Tim has shown quite clearly that it's possible to over-ingest water. You can probably over-ingest carbohydrate in situations where it's not required, and that's potentially detrimental. And if you look in the wider community health, some of the meta-analyses, the epidemiological studies, that there are hazards associated with high fat diets and too low carbohydrate. There are equally hazards associated with too high a carbohydrate. And I think the other important thing, again, I'm not a nutritionist, but it's, we focus on nutrients, but it's food, which is, so is it animal-based fat? Is it plant-based fat and protein? Are they refined and ultra-processed foods and carbohydrates? Are they, some of the more, less processed complex carbohydrates, so-called complex carbohydrates? So it's a complex space, and nutrition is, the history of nutrition advice, not just in the exercise area, but in more general health, is, science changes over time. It's iterative. It's, the very nature of science is about hypothesis testing, refutation, replication, and things change. But in the Twitter universe, immediacy and definitive answers are what's required, and often it's challenging to do that. So exactly. So maybe I'm sitting on the fence too much there, Glenn, but
no.
That's my sense, at least. So.
No, I agree. I mean, I had Javier Gonzalez on, and we were talking about how, and also Louise Burke, how going the way of the carbohydrate. Some people think you've got to have carbohydrate before exercise, during exercise, after exercise, every millisecond. But it depends. What are you training for? How often are you training? If you're training like once a day, do you really need to have carbohydrate every millisecond? And I think the answer is probably not. Yeah. Remember all the trouble we had trying to get glycogen low during my PhD?
Yeah, and some of those, and I think the other thing that's happened is, there's always been an appreciation of the individual. There's a famous quote, I think an English physician from the 1700s who was very successful, and it said it's not the disease the patient has, it's the patient that has the disease. And so whatever tools that this person used in the 1700s to personalize his management approach to patients, he obviously was quite successful. I believe he made a bit of money, and he actually invested it in research back then. And so the notion of personalized nutrition, personalized exercise prescription, and again, there's a balance between, the large cohort studies, the randomized control trials is the hallmark of evidence, if you like. But we've always got the outliers, we've always got the non-responders, the responders, okay. And for whatever reason, their biology is a little bit different, and their response to an intervention is a bit different. And I guess that's part of the wonder wonder of the human is, we're all distinct in our own ways, and so there'll be some general things that you can say. Well, exercise is good for you. I think there's overwhelming evidence that exercise or some type of exercise is good, but there'll be some people where you might have to play around a bit to find out which type of exercise might be the best for them.
Yeah, so it's
I think
individual.
it's similar with nutrition as well. So
yeah, because I think that's part of the problem with Twitter, because someone will read, oh, you need to eat 8 grams per kilogram of carbohydrate to maximize your glycogen. Oh, okay, so I better do that. But what exercise are you doing? Oh, I go for 30-minute run every second or third day. Or the other way, oh, I want to maximize my fat oxidation, because of this and that. But what are you training for? Oh, an 800-meter race. It's like, well, you're going to be using almost entirely.
It is. It is. The context is always important.
So why don't we just talk a little bit, because I just remembered that. So remember when we did that study during my PhD, we were trying to look at high glycogen versus low glycogen. And I just always remember that we depleted them the night before. We put them on 25% carbohydrate. We get them back in the morning and they were normal. Glycogen was normal.
Relatively normal, yeah.
Oh, no, sorry, that's not true. We did 48 hours. Sorry, we depleted them and then 48 hours on 25%.
We did a couple of interventions, yeah. In hindsight, maybe we should have done some arm exercise to send the glucose somewhere else. But it was, we ended up, it was interesting. You recall we had that, we had the 4 different levels of glycogen, and 2 of them were, I think one was 24 hours on the high carbohydrate diet, and the other was 48 hours on the low carbohydrate diet, and they had essentially the same glycogen, which was interesting because we ended up, you could start to separate perhaps the glycogen effects from the dietary effects, which is always the confounding effect in humans, certainly, where to manipulate the glycogen, you've got to change the exercise or the diet, and that then has systemic effects. I remember, I think you may not have been around when Jean-Marc Lavoie visited for a little while from Canada. And he's got some interesting studies where he's tried to deplete muscle glycogen, provide carbohydrate, manipulate liver glycogen versus muscle glycogen. And there's some complex interactions there that are hard to unravel in an intact human. But it was... and of course, in that study, we were looking at the links between glycogen and glucose uptake. And we had some permutations and combinations where we could try and unravel what was due to muscle glycogen per se and what was due to the background diet and systemic hormones and metabolites. I think Mark Hargreaves, when he was in Copenhagen, I think did some work on the single leg extension model where he's able to do that a bit as well. So, but that's one of the challenges and the frustrations of working with in a human model is all of the things that you can't always control. So,
well, it was frustrating, but as you said, it worked out really well because we were able to look at the effect of different glycogen levels on glycogen breakdown, on glucose uptake. But the main thing I was trying to get out there, I guess, is that people that are obsessing about wanting to have maximizing their glycogen or whatever. We just had, a hard time actually getting the glycogen low. We had no problem getting it high, but so just, it ended up normal, even if you ate sort of no carbohydrate hardly.
Well, you could argue perhaps that, if it's well protected, its resynthesis, as you know, is preferenced in recovery over the liver, which would suggest to me that, there's been obviously some level of pressure to ensure that there's a bit of glycogen around in muscle, which perhaps speaks to its importance for muscle function. So
exactly right.
But not to overstate that, but and look, I think if you're going to be in an event where your glycogen availability is likely to be limiting, then the strategy would be to try and maximize its availability and potentially maximize its utilization. And there's no precise way of getting that exactly right. We can't look at your smartwatch and it can tell you what your what your glycogen concentration is. I mean, I've seen there's stuff out there that people should, having continuous glucose monitoring during exercise, and titrating their carbohydrate intake to that. I think that's probably a little. I'm not sure there's good evidence for that sort of precise regulation, but you can sort of get some approximations without necessarily knowing exactly what your blood glucose level concentration is.
Exactly. Now, another thing that came out of that study that was interesting was, well, okay, the first thing was that, and Eric Rector had kind of shown this in rats, that if you have high muscle glycogen, you use more of it, right? And then, so it's like you almost like waste it. And that's one of the chats we had, and I just wanted to talk about that again. When we did that study, they were exercising at 65% of VO2 max for 40 minutes. And, they were able to do it on the low glycogen, on the moderate glycogen, and on the high glycogen. But on the high glycogen, they were like using much more glycogen. So you start saying, well, hang on, why are they actually doing that? How is that helping the organism to survive and reproduce, to sort of waste glycogen? Do you have any thoughts on that?
Not really. I mean, I think it could be as simple as, the sorts of dynamics, the substrate availability and the factors that drive the activation of glycogen phosphorylase. I mean, Eric showed that when glycogen is higher, phosphorylase is higher. There's this general, if you consume more carbohydrate, you tend to burn more carbohydrate. If you consume more fat, you tend to burn more fat. At a given, subject to the intensity at which you're exercising. Obviously, if you go to a higher intensity, that dynamic is perturbed a little bit. So, it's similar to the work we did with Mark Hargreaves, exercising in the heat. Again, that intellectually that had its origins with my time at Ball State, where Dave and Bill Fink had done some work on heat and glycogen metabolism, and then Doug King, who was doing his PhD, was around acclimatization, muscle glycogen use during exercise in the heat before and after acclimatization. And, the work that Mark did subsequently, you burn more glycogen during exercise in the heat, probably because plasma adrenaline is high. We know that activates glycogenolysis. It activates, or it's associated with an increase in PDH activation. We did that with Lawrence Spreed and Matt Watt, probably because of the increase in glycolytic flux. An increase in muscle temperature plays a role. But when the subjects fatigue prematurely in the heat, it's not because they've run out of glycogen, even though they used more glycogen. There were other factors contributing to that, whether it was cardiovascular limitations or temperature limitations in the central nervous system, whatever it was. So, as you know, fatigue is a complex phenomenon with lots of things, and all the studies we've done, the subjects still fatigue even though there's plenty of glucose there.
Exactly.
And maybe the glycogen's not in the right spot, and we don't have the tools for measuring where the glycogen is. And I think Niels Ortenblad in his morphological studies, trying to look at site-specific glycogen depletion, have shone an interesting light on the criticality of glycogen. There might still be at a whole muscle level enough glycogen, but at the T-tubule or at the SR, it may be limiting where the ATP is really needed, and that could be the limiting. Now, of course, I've grown up, studying metabolism, so I have a metabolism bias, but it could be something entirely independent of glycogen that's causing the fatigue.
Exactly, and this is one that's again, this was in my PhD, we were like scratching our heads about this. So, and I've brought this up with Jorgen Janssen and with Javier Gonzalez, saying, why do you think people fatigue? So just to make sure people are on the same page. So what we're talking about here is if you're exercising, say, I don't know, 70% VO2 max continuously for as long as you can go. Eddie Coyle showed in 1986, that the glycogen goes down and then it doesn't really change, but you keep going for another hour if you ingest carbohydrate. And, the carbohydrate oxidation is still, fine. The glucose levels are still fine. Nothing appears to happen to the glycogen, but as you say, there might be compartmental changes.
Mm.
Then they fatigue. Because, remember we even did the study with the biopsies, at the point of fatigue and tried to see if anything changed, in the muscle in terms of what we looked at, and you couldn't see it. So I was going to ask you about that. Yeah.
You remember that the IMP study where, the IMP is at least one marker of ATP breakdown products, was sort of similar at the point of fatigue, even though there was a 60 to 90 minute difference in time to getting to that point. So, but again, that doesn't in any way exclude other factors outside the muscle that are contributing to the fatigue. I think the crosstalk, either both through bioactive molecules that are floating around in the bloodstream, or neural connections, the various reflexes which act to protect the organism. So if you think about hypoglycemia, hypoxia, hypothermia, and the effects that they seem to have on central motor drive, it's it's a good there are there seem to be some reflexes which are protecting the organism when some of those crucial parameters are being challenged. So
yeah, and Javier and I were also sort of, he had this idea that he thought maybe it was coming from the liver, and I know we talked about that somehow, some sort of, because you mentioned the double tracer study, so I don't know if you want to talk about that, but the fact that you're sparing your liver glycogen, I wonder if there's some sort of signal or something that enables you to go longer. But again, why do you fatigue? So I don't know if you want to talk about the double tracer study, because that was a great study. Yeah.
Yeah. Well, I mean, we as you know, we looked at the whole body glucose kinetics with a stable isotope, and then we had the ingested glucose labeled with a radioactive isotope. Others have done it with a different stable isotope, and Asker Ukendrup did those studies much more extensively than we did with some of those. He was able to look at oxidation as well. You'll recall we used a pretty high glucose concentration. In hindsight, maybe we should have used a bit lower, but the appearance of the glucose out of the gut began to level off, and at that point, the endogenous glucose production began to increase. So there's this interesting regulation around splanchnic, if you like, the sum of the liver and the gut. It's certainly when you ingest carbohydrate is regulated in some way. I'm sure David Wasserman would probably have some views on that. And it's similar after exercise, where there's splanchnic escape of glucose that's ingested to allow muscle to get hold of that carbohydrate. So
yes, this comes back again to what you said earlier, that so even if you're not having that much carbohydrate, it tends to go through the liver and gives the muscle the first shot at it, which, as you said, does sort of suggest that the muscle carbohydrate metabolism is important. Now, you're saying there, we had a couple of questions on Twitter, and again, it came up with Javier Gonzalez about the amount of carbohydrate you ingest. So, we back then, it was sort of like a gram a minute during exercise was sort of, so 60 grams an hour was what we thought was sort of maximal. I don't know if you've kept up on this literature, but, even back with the Oscar there, years ago, looking at fructose versus glucose, and getting right up above 90 grams an hour, 120 grams an hour.
I mean, we did a long-term study with Damien Angus, and I think in terms of carbohydrate utilization, or the potential, it was up 1 to 1.3 grams, and how much can you get out of the gut? And so the balance there is, well, how much do you need to deliver, and how much can the muscle take up? So, I think if you look at the dose response studies, and there's a few of them in the literature looking at different levels of carbohydrate, and we've got some unpublished data from Damien Angus's PhD that's still floating around. We actually mapped or graphed the rate of carbohydrate ingestion and endogenous utilization, and at about 4%, which is 40 grams per hour, if you assume about a liter of ingested fluid. in the fluid form at least. That's where below, if you went below that, you had to draw on your endogenous liver stores of glycogen to maintain your RA. If you went above that, then you're able to blunt the liver mobilization. So I think you probably need, whether you need 90, and look, I think USCA's studies have shown that if you can do interventions that increase the bioavailability of the glucose, that is associated with increased performance in the protocols that they've used. There's going to be a limit eventually where the muscle may well be the limiting factor. GLUT4 unlikely. I mean, the translocation to GLUT4 is pretty complete. So then the metabolic factors, you've got glycogen depletion. So there's a fair drive for glucose diffusion into the muscle under those conditions. And then we come back to where we were talking earlier about, well, you've still got glucose availability, but you still fatigue. So is it a critical amount of glycogen in the key locations in the muscle, or is it something outside the muscle? Is it liver glycogen depletion? I mean, you'll remember that timing study we did where we did 7% throughout exercise, we did no carbohydrate and then a big whack of
0.21,
a very, very concentrated carbohydrate, which could have stayed in the gut, but the glucose level went way up, but they still stopped. So was that because the liver was signaling to the brain, hey, I'm out of glucose, out of glycogen, you need to stop, or was there something else going on?
Yeah, that's the one I was just about to say, the old 0.21. Yeah, so they either had, 2 hours at 70%, these are trained people.
Yeah.
And then a
7%
trial
every 15 minutes, and then a time trial. Yeah.
Or they had 0 for the first 90 minutes.
Yeah.
And then 21% for the last bit. And we thought, because, Coyle had shown that, and you had shown also in 1998, that carbohydrate ingestion during cycling doesn't affect muscle glycogen use. So we thought, okay, so why don't we just try and, back end it, get it all in there. It's not going to affect glycogen anyway. So we end up with more glucose availability. So as you said, the glucose was just went way up above the 7%. The insulin went up, so it should have been driving glucose uptake, but they did not do as well as when they had 7% throughout. So that was as you said, that was another one where we were saying, well, maybe it's the liver depletion of glycogen or something.
Yeah.
Now, just another one is this thing where people think, I'm going to the gym, I better have my sports drink or whatever. Again, this concept of, you've got to think about, is it going to affect your performance or your and so often if people go into the gym for 45 minutes, an hour or whatever, what do you think about that? Carbohydrate loading, carbohydrate ingestion, it seems like maybe an hour, anything an hour or a bit around there, it's probably not going to make much difference.
I wouldn't have thought so. And you need to pay some attention again to what is it that you're doing. So how hard are you exercising, and you're likely to be limited by carbohydrate. When this first came up, the whole question of whether carbohydrate in the mouth, and the whole sort of mouth rinse story, and that has an interesting. I mean, there's an old paper in JAMA on the Boston Marathon where I think in 1924 they measured, various biochemical parameters, including blood glucose after the race, and there were people who were exhausted and feeling, faint-headed and nauseous. And then the next year they repeated the study at the Boston Marathon, but gave barley sugar. And I think there's a quote in the paper that as soon as I put the candy in my mouth, I felt better. So and then there's good evidence now of links between carbohydrate in the mouth and the central nervous system. So there may well be other factors. Having a little bit of a drink of carbohydrate might just make you feel that little bit better. And if you're off to the gym after an overnight fast, your carbohydrate stores may be down a little bit, and just having a drink might make you feel a bit better. And we know the central nervous system is very important in the activation of muscle and how you feel. So there could be benefits beyond the metabolic. But I think my sense of the glycogen loading literature is that below 60 to 90 minutes, there's probably not much benefit, and it's 90 minutes and beyond where you see. But again, intensity. I mean, there are some repeated high intensity. Paul Balsam has shown that glycogen loading can improve performance for the sprinting efforts. And I think Niels Ortenblad recently had some work on high intensity performance, and again that localization of the glycogen, particularly if you're performing bouts that are very demanding on the sodium potassium regulation and the calcium regulation. There'll be a high demand on the glycogen stores around those ATPases. It may well be a factor. So
you're talking about repeated sprints there, or? Yeah,
it was came out in MedSci just late last year. These repeated high intensity efforts, and again using his morphological analyses to look at where glycogen was located in the muscles. So
I guess again, it's, as you've said a couple of times now, if glycogen is limiting, then you'd expect the performance. So as you know, the classic Hermansen 1968 or whatever it was, 30-second sprint, Wingate on the bike, they use 50% of their glycogen in the 30 seconds. So I guess if you're going to do those continued sprints, the glycogen could become low enough. Is that what you're thinking? Or
look, and there will be other factors. I mean, the repeated high intensity efforts, looking at things like other metabolic changes, acidosis in the muscle, changes in SR calcium function, as well as glycogen availability. We've done, I think Lawrence Spreet's done some work on that. We did some work with changing the recovery duration between, or allowing some things to recover and some things clearly didn't recover, to see if we could uncouple glycogen from other changes in the muscle. But it speaks to the complexity. I think we're very naive if we think that there's a single mechanism that contributes to fatigue. It's probably a multiple, and depending on particular circumstances, there's going to be one will be more or less important depending on the context.
Yeah. So as you said, exercise in the heat, there's the classic studies. They showed if you exercise to exhaustion 10 days in a row, you'd sort of get up to the same temperature. The studies in the heat chamber. So as you said, you might be using more glycogen, but it's not. It doesn't mean it's causing fatigue. It's probably more like central.
Yeah, potentially.
Yeah. So what about after exercise? So I saw someone on Twitter, was Raul Saldren, said after a hard workout, if you're doing very low intensity exercise, do you think, and you're ingesting carbohydrate, do you think the body could actually resynthesize glycogen? I guess it depends how low it is, but not sure if anyone's looked at that.
I mean, there has been, during recovery from exercise, maybe more intense exercise, there's suggestions that glycogen can be resynthesized from lactate. And of course, lactate can be oxidized. And so that's an alternative. So if the interactions between lactate utilization, glucose, there may be some glucose that can be. I think I'm trying to remember wasn't there a student in Dave's lab? Was it Choi who did active and passive recovery on glycogen resynthesis?
Oh, I don't know. I remember the classic, talking about lactate removal, passive versus active, but I can't remember that one. I remember the J. Mark Davis doing the intermittent exercise, and he'd be finding you'd get some glycogen reduction in glycogen breakdown, maybe resynthesis during the recoveries when you drank carbohydrate. But I'm not sure about this.
Yeah, I just looked the great advantage of being online simultaneously. I can look up Google. It's effective passive and active recovery on the resynthesis of muscle glycogen. These data suggest that the use of passive recovery following intense exercise results in a greater amount of muscle glycogen resynthesis than active recovery over the same duration.
Mhm.
So they did, what was their active recovery was 30 minutes at 40 to 50% VO2 max. So I guess it's all about the balance between substrate utilization and substrate availability for glycogen resynthesis. And depending on that interaction will determine how much glucose is available for glycogen resynthesis and how much of it needs to be utilized for oxidation. Recognizing that during the recovery period, you tend to rely a bit more on fat oxidation during recovery. Again, that argues for, at least in the non-exercising and the unfed state, preferencing the resynthesis of glycogen rather than the ongoing oxidation of carbohydrate during recovery. But yeah,
because the intensity is so low. Again, you've got
Interesting.
to get that glycogen in there because it's so important. So if you're going so really slowly after exercise. So again, if we think, we're hunters and gatherers, these days that we can choose to do this exercise have been quite short. But, if it makes sense, if a tiger runs out while you're picking berries and you have to sprint to a tree or a couple of trees and you use glycogen, and then you're like walking later, it would make sense that during that low intensity exercise, you'd resynthesize glycogen, right?
Yeah. Look, I'm not across the literature. There's been a little bit of stuff on, metabolomes in humans and chimpanzees and gorillas, and the muscle and looking at the muscle and the brain metabolomes and how that's evolved. But and that whole sort of persistence running theory, and with a lot of focus on thermoregulation and control of muscle function. The metabolism story is an interesting one, maybe to explore a bit further. And evolutionary biology normally has some pretty good insights into trying to explain why things have ended up there. There are advantages in particular adaptations that have occurred over the years. But I'm not across that literature in detail, if there is indeed any. So.
Okay, just another question from Twitter, and it fits with something that actually Tim Noakes again sent a few weeks ago before Javier Gonzalez came on. He was sort of saying that, it was sounding like he was thinking it was potentially, dangerous and bad for your health to be packing in carbohydrates during exercise. So, we're talking about 60 grams, 90 grams. Some people are saying 120 grams. Where does it go, I guess? Can it be damaging that, if you haven't used it during exercise, is it going to mess up your homeostasis after exercise? So the question here was, it's Nanette Sousa. What are the health implications, if any, of consuming 120 grams of carbohydrate an hour during exercise regarding inflammation markers, dental hygiene, endothelial wall damage through inflammation? And I guess, and it also reminds me, remember that when we did the double tracer, we couldn't account for, remember we tried to work out how much of the glucose that they ingested actually showed up in the blood. So I guess if it's not getting absorbed, is it, so do you have any thoughts on that? It's a bit of a tricky one.
Well, look, I think, the general principle of anything in excess is, needs to be considered. If it's excess to needs, it's going to be, and I think there's some work, I think even out of Eric and Benter in Copenhagen, excess carbohydrate as well as excess fat had some negative effects on insulin sensitivity, but via different mechanisms. And, as I said, I'm not a nutritionist, but, some of that meta-analyses work that Walter Willett has done, who, he has argued that fat has perhaps been unfairly demonized in diets, and one would have to say that a focus on low-fat diets at a population level hasn't prevented metabolic diseases and obesity. So clearly it's more complicated, and it goes back to foods and the source of foods, the types of foods, the source of the fat, the source of the carbohydrate, the type. Is it processed in various ways? So, I think excess either way, and there's a lovely graph in that, I think it was in Lancet Public Health, where they pulled together all these studies, and the hazard ratio for for premature death was high below 50% carbohydrate, and not the slope wasn't as steep, but on the high carbohydrate side, it still went up. So I think anything to excess is probably going to have consequences, and it needs to be considered. And so again, the challenge is, well, we don't have carbohydrate meters on our body. What are some reasonable estimates that we can make about rates of carbohydrate oxidation? You can make some reasonable attempts from the literature about how much carbohydrate you're likely to use. And if you're not going to use 120 grams per hour, then there's not much point ingesting that much, I would have thought. But there's probably benefits of ingesting some, because it's not zero utilization of carbohydrate. So
yeah, and the other thing, I guess, if we think about it, is ingesting, the studies we did and others have done, ingesting glucose only, ingesting fructose only, ingesting, infusing intralipid, all sorts of things people have done. They're not physiological. They're not food. They're not what we've been, adapted. Again, I'm back to evolutionary, but you know what I mean? Like, I think often, I've mentioned it a few times in this podcast, Michelle Kesky from Deakin, showed that if you do an oral glucose tolerance test, so just drinking 75 grams of glucose, you actually don't get the normal increase in microvascular, so capillary sort of blood flow that you get when you have a meal. So, and it's probably because it's not normal. It's not normal to just have, when are you ever going to have 75 grams of glucose and no fat, no protein,
well, if you're an endurance athlete on a bike,
exactly,
that's probably, you'll be drinking, all sorts of, glucose, sugary drinks was probably an example, but most of the meals would be, and those data from the Tour de France, where a lot of that energy intake was consumed as, high carbohydrate, high energy intake during the race, over the 24, those 24-hour studies. So, but I think, there was Jeff Horowitz and Ed Coyle had the, if you add fat to glucose ingestions, which is a bit like, what you might see in a meal, you get different responses than if you have, single nutrients. So not surprising. I think isn't there an old study about caffeine and coffee eliciting slightly different metabolic responses? If you take caffeine as pure caffeine, or you take a coffee which has other ingredients, you get subtly different responses. So
yeah. So I mean, I think they work. If you have glucose or whatever on their own, they work. But then they're to improve performance. But then you wonder what's happening after the exercise. Is it, as I said, it seems to have funny effects on blood vessels that you don't get with a meal. So wonder if it's just basically, as it's like you said earlier, having ultra processed foods and whatever. It's you're probably better off having food if you can. So if you're not doing, say, more than an hour a day of exercise, why have these sports drinks, or why have glucose and fructose, or just have a meal, just have food?
Yeah, and there are analogies there with sodium. I think Ron Morn has shown that if you don't need rapid rehydration, you can achieve pretty good rehydration through ingesting fluids and ingesting meals, which for most people normally have enough sodium for what we need. But if you're limited, your appetite is suppressed in some way, you don't eat and you need some rapid rehydration, then upping the sodium content of the beverage helps you, rehydrate that a little bit faster. If you need to rehydrate quickly, if you don't need to rehydrate quickly, you can take a bit more time and do it in a slightly different way. So
exactly. Actually, this also reminds me of again on Twitter, a lot of talk. And if you just talk to someone on the street and they say, "Oh, what about electrolytes? What about electrolytes?" And it's almost like the focus is nowadays sometimes on electrolytes rather than the carbohydrate content of a drink. So, during exercise, so again, remember the classic study we did with David Kostor. So I responded to someone's tweet the other day saying the amount of sodium that's in a sports drink is so low, because if you put it higher, it's unpalatable. But also during exercise, your sodium tends to go up when you're sweating. So I don't know. Do you want to just explain what I'm trying to stumble over here?
Well, yeah, I mean, you'll recall the study we did with the different levels of fluid intake. And interestingly, when we gave roughly half of the fluid that was lost, this plasma sodium stayed at pre-exercise levels. And so, what's regulated? Plasma sodium is an important regulatory factor. They still lost a bit of their plasma volume there relative to when we gave them full replacement, but that actually their plasma sodium went down a little bit. So, I think certainly electrolytes have a role to play. And if you look at the oral rehydration solutions for diarrheal disease, they tend to have a bit of glucose and a bit more electrolyte to help retain some of that fluid where you've got large fluid losses induced by the disease. So again, a little bit's important, but do you need to have as much? Possibly not at the extremes. So
okay, so you think having these like tiny amounts of electrolytes in sports drinks during exercise is actually having much effect? Because I was thinking it was too small to have an effect. I thought that's what we worked out with the Coastal study, that when you add sodium, the amount that's in sports drinks, you can't even pick it up.
Well, I think you're referring to the study we did when we had different levels of sodium to look at deuterium, the absorption of water.
Yeah.
Yeah, that didn't make much difference at all. I think the important thing is in terms of retention. The urine volumes tend to be a bit lower if the sodium is a bit higher. I know Ethan Nadell always used to argue that some of the electrolytes in sports drinks should be a bit higher, particularly for the longer type exercise. But again, that's that balance between fluid intake and whether you need to add electrolytes. And you still get some of the hyponatremia that Tim has described with excessive fluid intake, even if there's a little bit of sodium on board. So there are these complex interactions that are taking place around ingestion and bioavailability, and then excretion and retention.
Exactly. And again, I guess it's sort of like saying everything in moderation, but also, as we talked about with the carbohydrate, do you need to ingest carbohydrate or have high glycogen? It depends on what your needs are during the exercise. It's the same with the fluid ingestion. I had a friend during my undergrad, and he did an Ironman race, and he said he urinated 8 times during the bike. And he said, "What do you think that was?" And I said, "Oh, it's because I think you were drinking too much." Because he was doing, had this preconceived idea of 250 mils every 15 minutes, irrespective of the fact it was a cold day, and he was losing heat, through convection.
Yeah.
So, that's when you can get the hyponatremia, right? When you're drinking more than you need.
Yeah, and I think that's an important part that comes out of there are the debates and arguments in the scientific world, that the subtleties and the nuances of that are often lost when it's translated into the wider world. And I think just making people aware that there are potential hazards with excessive fluid intake, potentially excessive carbohydrate intake if it's not warranted on a regular basis, and people need to be mindful of that.
Yes. Okay. So I'll just go back to one more Twitter question. I think we're getting a bit low on time here. Jonathan Stein asked, role of glycogen depletion in leading to AMPK activation in the liver and muscle.
Well, you're the expert on AMPK, Glenn, so I should hand that over to you. But it's interesting that the glycogen depletion, it's been shown to be when it's acutely manipulated, it has an influence on AMPK activation. And you're a co-author on a recent paper looking at the links between the extent of glycogen depletion and insulin sensitivity, and the work out of Copenhagen would suggest that the activation of AMP kinase may be an important component of the increased insulin sensitivity after exercise. But the glycogen depletion wasn't a major predictor of the increased insulin action, even though you might think that that's an attractive mechanism. If you're going all the way back to the early Bergstrom papers with the glycogen depletion, glycogen supercompensation, and the suggestion that maybe there's a local factor related to insulin action or glucose uptake. So I think there is a link between the extent of glycogen depletion and the extent of AMPK activation, although you uncoupled that.
Yes, I did.
with your training studies. So again, there are some complex interactions there that we still don't fully understand. Everything that's going on there.
All right. Now, I meant to say at the start, but you've had changes in your career direction. Why don't you tell us a little bit about what your main position is now? And I don't know how you manage to still pump out papers.
Well, I don't. I haven't really published original. I've published a few reviews of late, but I guess that's what you do when you run out of data. And you're still occasionally invited to do some things, but living off past glories. We've got a little bit of data. We've still got some unpublished data on GLUT4, which we're on GLUT4 regulation, which is we're hoping to write up on, working on it at the moment. But in the last few years, I've got more involved in institutional research leadership roles. Initially in a part-time role, but I was dean of the medical faculty for 12 months, just filling in, and then after that, that sort of finalized, if you like, my transition to institutional leadership, and I've been a full-time pro vice chancellor in the research portfolio here at Melbourne. Acting deputy vice chancellor research for a period of almost about 3/4 of a year when our DVCR was the acting provost. So, and that's been it's a very stimulating and rewarding job. You grow up focused on your own research and those of your lab and your students, and then you end up in leadership positions either as a head of department or as a dean or in chancellery, and you thinking about the overall research enterprise, not just your own research enterprise, and that's been very rewarding and satisfying. So
it's been amazing though. You keep your eye on the literature. You're always on top of the literature. And the other thing I meant to say at the start as well is you always had a photographic memory. So I'd go to see you and you'd pull out a paper and you not only knew where it was in your pile, but you went straight to that paragraph.
It's deteriorating, I can assure you. I think probably age is catching up on some of those things. But I think fundamentally, the reason I think we all get into this is we're interested. The thrill of discovering something or getting an insight that trying to understand things is still fundamentally, I think, what motivates all of us, the intellectual curiosity, and I don't think you ever lose that. Your ability to keep on top of that is determined by obviously what you're doing, and you make conscious choices. In various points in your career, you keep doing various things or you do other things, and that. But I think fundamentally, you'll always be interested. I always see what's coming out in the literature. I still get the thankfully now you get it electronically rather than having to look through the old current contents book. So there are ways that you can keep on top. I wouldn't say I have a deep understanding of all of the contemporary literature, but I try and at least see. And if I get a bit of time, then I can go and read something in a bit more depth. So
I remember the good old days. Well, the old days before this was available, when we used to go down every Friday to the medical library. And Jay, Journal of Applied Physiology is here, and it'd be like 2 months because it'd be coming on like a boat from the US. Okay, do you have any time to? Because I mean, GLUT4 we haven't talked about, and that's been your big
Yeah, look, I'm happy to keep going a little bit longer. I mean, I think one of the interesting parts of the journey is, you know, the increased application of various techniques that have evolved in you know, molecular and cell biology, and you know, we haven't, you know, we've applied them to the extent we can to questions in the human model, but you know, obviously the last few years most of the research interest has been on the regulation of GLUT4 expression. And that really grew, you know, as an extension of, you know, we started studying glycogen metabolism, then we gave people carbohydrate, which was, you know, influenced muscle glucose uptake. And of course, Mike McCoy, who was the PhD student around the same time, was studying GLUT4, and you remember he was doing, you know, the I-125 autoradiographs for GLUT4 protein expression and
labeled insulin, yes,
and different and in different, you know, subject groups. So, you know, I think we've done, at least to the extent you can in human models, try to identify some of the mechanisms by which exercise increases GLUT4 expression in muscle. You know, there's some interesting ways in which you know, a single bout of exercise and then training increases GLUT4. So that's been the focus in the last few years, the last, you know, 2 decades, I guess, as active research. But as I said, we've still got some trailing data that we're just looking at the moment around histone modifications and the role of P300 and CBP in that process. So, yeah.
So regulation of how acute bout of exercise increases GLUT4, the mechanistic level.
Yeah.
And also, remember we looked at, no, sorry, it was you and Sophie Yeo, I think I might have been on the paper, but looking at acute exercise and GLUT4 and fat as well.
Yeah, we, Sophie did that, particularly in the type 2 diabetics. Marcelo Flores-Apazo, who's back in Chile now, is my last PhD student, because I haven't taken any on for the last few years, given my other commitments. But and we saw in the diabetics who have a lower GLUT4 in adipose tissue, and that's been shown previously in humans, that exercise increased GLUT4 in adipose tissue. But when we look at in normal, in healthy young volunteers, we haven't really seen an increase in GLUT4 or in mitochondrial markers. So this sort of notion of you know, browning of adipose tissue, I think perhaps in our hands at least, not as apparent in humans as maybe it is in the rodents. But it's interesting. A lot of that was driven, as you know, by work from Barbara Kahn, suggesting that you know, reduced GLUT4 in adipose tissue was an important contributor to insulin resistance, whole body insulin resistance. David James recently had a paper in Cell Metabolism where he looked at 30 different strains of mice and couldn't really see a consistent relationship between adipose tissue GLUT4 and insulin resistance. So I think that story is probably a little more complicated, as it always is. You know, the role of GLUT4, certainly it's important in muscle, and both for acute exercise, but also for some of the exercise adaptations, and may well be important in adipose tissue certainly as, you know, getting glucose into the adipocyte, but also in terms of some of the signaling that might occur from adipose tissue. So, but we've only done a couple of studies in humans in the adipose tissue, and most of the focus has been on the muscle. So,
yeah, and it's interesting that people would tend to assume that people with type 2 diabetes would have lower GLUT4 in their muscle because they've got less glucose uptake, but it's pretty much normal, isn't it?
Most of the studies would suggest, there's a couple of studies here and there where GLUT4 expression has been a bit lower, but it generally tends to be about the same in muscle. Interestingly, you know, there's 3 or 4 studies in looking at adipose tissue, and it's consistently down in the adipose tissue in diabetics and people pre-diabetics, people with obesity. So there could be something interesting there. Still need to work that out. So.
I guess I should have said at the start, so GLUT4 is a glucose transporter that's insulin regulatable. All right. Well, thank you very much for your time, Mark.
No, thanks, Glenn. Thanks for the opportunity. You've been very busy at speaking to lots of colleagues and friends in the series. So I hope there's some interest. And thanks for the opportunity to catch up. And
there's been a lot of interest on Twitter. When I put it out there, lots of likes, lots of people excited to hear you talk. Is there any way you can give like 2 or 3 bottom line takeaway messages from this talk?
Well, I think carbohydrate availability, particularly muscle glycogen, is important if you're going to exercise hard. And that's not just necessarily elite athletes, but the weekend warriors who go pretty hard. So it's important for the intense exercise. But that needs to be, the caveat on that is that if you're exercising, some of the recommendations that you might see might not always apply to you as an individual, depending on how hard you're exercising, how long you're exercising, how long you've got to recover, all of those sorts of things. I think you can alter carbohydrate metabolism through changes in availability. And so that's where diet and nutrition has really played a role and whether, I think, given the complexity and the multifactorial nature of performance and fatigue, not surprisingly, you can often induce large changes in metabolism that don't always result in meaningful changes in performance. Yeah. Which speaks again to the complexity of human performance. I guess I would try and everything in moderation, including moderation, as they say. If excess, the body will deal with excess nutrients, and excess carbohydrate will be dealt with in a way which may have some downsides, and you need to be aware of those. And excess fat may have some benefits, but there may also be some downsides, and just being aware of that. And I guess the other important thing is that we don't always have the answers as quickly as our colleagues on Twitter might want us to have, and that's the nature of the scientific process. And views change, and some of us may need more evidence to change our views than others, because of our inherent biases and prejudices. But the immediacy of Twitter is not necessarily well calibrated with the nature of the scientific approach. So I guess that's something we just have to live with. So,
well, beautifully put as usual. Very clear, articulate. And I might just finish up. You mentioned Marcello. Marcello sent a nice message, and given he was your last PhD student, well, is there any way you might take another one on? Is he officially your last?
Look, given my current roles, and Marcello will attest that I used my network of friends and colleagues to help support Marcello during his doctoral work, given the other commitments that I had. So in my current arrangements, I can't say I'm always happy to have conversations with students who are in the department, and but I don't think I'll be taking on any students as a principal supervisor. So he will be, I think, pretty confidently say he'll be my last PhD student, of which I was the principal supervisor. So.
All right. So as your second PhD student, this is from your last PhD student, Marcello Flores Apazzo. Excited to hear Mark's podcast. He was my supervisor and still is my mentor. Amazing human being. And I'll back that up as well. You've been great with me, mentoring, etc. throughout. So thank you very much.
Well, thank you. And I've got as much, if not more, from my students than they've got from me. So it's always been a pleasure to work with talented and motivated people like yourself and Marcello and others. So, yeah.
It sounds like your farewell. It's not meant to be like that. Good on you, Mark. Thanks again.
All right. Thanks for the time, Glenn. We'll catch up in person.
Yes.
We'll do that.
I'm going to give you that book. Okay. See you, mate.
Bye.
Bye-bye. I hope you enjoyed this podcast. Please like, subscribe, pass it on to your friends and colleagues. Check out the other podcasts. Thanks again.